All right, friends, again, another lab that was done reasonably recently is Lab 9 on transpiration. We did the whole plant method of this lab, and there's another method you can do as well, just don't worry about it. Please remember what transpiration is, the loss of water through the leaves, and the control of that is maintained by stomata, the holes with the guard cells that may open and close. Please remember the importance of cohesion to that process. The water molecules pull them each other up as one leaves the leaf, as we can see here, as a water molecule leaves the leaf because we have a higher concentration of water in the leaf than out. It leaves via osmosis and pulls the other water molecules out with it due to cohesion. We tested the effects of various environmental factors on how fast transpiration occurred or how much transpiration occurred in a given time. We tested humidity, wind, light, presence of light on all of these on transpiration. We did this by massing the plant before and after. We covered the root ball in plastic so no water could evaporate from the surface of the root ball. It had to leave through the leaves only. So that's how we did it. We masked before and after, and of course there's our wind. Remember our humidity treatment. We spritzed it and covered the plant in a bag, and we also exposed some plants to light. So you definitely should understand all of these concepts. Hydrogen bonding as it relates to cohesion, also adhesion, clinging to the xylem tubes. This is in one of your plant podcasts, a little note about how stomata work. When we want them to be open, when it's plenty of water in the environment, no drought, potassium ions from surrounding cells are pushed in to the guard cells. And of course, remember, water follows the solutes. There's a high solute concentration in this guard cell, relatively low water, so water would flow in by osmosis. Conversely, in a drought condition, potassium will be forced out, pumped out of the guard cells, and again decreasing the water out here, relatively speaking, and so the water will then follow by osmosis, leading to flaccid or closed guard cells. Okay? So what we found was that the transpiration was highest in as wind was increased and we had light, that transpiration was the highest compared to our control treatment. Transpiration was less in the humidity treatment. When we had high humidity, the transpiration went down because the concentration gradient from the inside of the leaf to the outside of the leaf was not so steep. If you'll remember, the wind we said was blowing the water. Here's my leaf, and the water molecules come out of the leaf. Well, the wind just blows them away, creating a perpetually low concentration of water immediately outside the leaf, and so the transpiration is higher. Okay, here's a pretty straightforward essay about this very lab, giving you data and different conditions. They want you to construct a graph, explain why the results showed up the way they did, explain the concept of water potential. Please, feel free to talk in terms of water concentration. High water potential, high water concentration moves to lower water potential, lower water concentration, and that sort of thing. All right, that's it for Lab 9. That's your quick reminder. Let me know if you've got any questions about that. I'll catch you in the next lab review.